Hello guys and welcome back to Excelsior Academy. So today we'll be doing the last topic in the DNA replication, and that's we are going to discuss about the telomeres and the telomerase. So before we talk about telomerase, first thing is to know about the telomeres. Okay. So guys, I hope you remember when once I was speaking about the red mechanism. Can you tell me about the red mechanism? I have mentioned once only. Yes. It is the replication end, the D loop mechanism. Okay, so just please remember. Okay, so the major function of telomere is to cap the ends of the chromosomes and protect the chromosomes from the red mechanism. Now, as cells divide, telomeres continuously shorten with each successive cell cycle. ये भी हमने discuss किया था, right? And telomerase, it provides the necessary enzymatic activity to restore and maintain the telomere length. So this is the second mechanism by which by which a cell is actually maintaining the length of the telomeres. Okay, what was the first mechanism we have discussed?
Using a protein instead of a primer and
Yes.
instead of RNA, and that synthesize the DNA.
Very good, very good. So that first mechanism is also known as protein priming. Okay, good. So the vast majority of tumors activate telomerase and only few maintain telomeres by ALT mechanism relying on the recombination. So what is ALT? It is alternate lengthening of the telomere, which is happening in the case of the tumors. Okay. So guys, कैसा होता है कि it's like, it's a signal. So if you keep on dividing, a cell gets a signal that after these many generations, let's say around 40 to 50 generations, okay, the telomeres get shortened, and at that time, the cell should undergo apoptosis. Okay, almost the telomere is gone, it's not present sometimes. So it's a signal to the cell to undergo apoptosis. But in the case of tumor, you have got overexpression of the telomerase. So क्या होगा? Every time during the DNA replication, the telomeres are also getting replicated properly. So they are not getting shortened. So the inhibition to the survival is prevented. समझ में आया? So the signal which should give that the cell should undergo apoptosis is now inhibited in the case of the cancer cells. Is this part is clear to you? Like how in the case of cancer the telomerase is acting?
Yes, ma'am.
Okay, so the telomeres and the telomerase are the attractive targets for the anti-cancer therapeutics. So in the case of like cancer drugs, you can have anti-telomerase drugs, like which is going to inhibit the activity of the telomerase. Okay, so you can design drugs like, since we know that it is RNA dependent DNA polymerase. Okay, so your telomerase is also known as RNA dependent DNA polymerase. Now, what kind of drugs, as a scientist, you can think of it? Like आप ऐसा क्या डिजाइन करोगे जिससे आप टेलोमरेज को इनहिबिट कर सकते हो? कौन से प्रॉपर्टीज को आप टारगेट करोगे टेलोमरेज की? थिंक लाइक ए साइंटिस्ट. सोचो.
Something that competes with the, something that also latches onto the OH group that the primer can provide, or something like that.
ना फिर से बोलना श्रुति.
Something that will compete with has the same kind of active site that telomerase has, and kind of binds and prevents it from binding to anything else.
So like kind of you are talking about ऐसे जो like in the case of competitive inhibition, ऐसा?
Yes. Yeah.
Since the target over here is the substrate over here is the DNA.
Yes.
Right?
So we cap the end of it.
Okay, you're talking about the end of it. Okay, thoda sa complicated hai, because then you need to design the drug also, which is having the structural morphology similar to the end of the DNA. right? So that synthesizing is quite difficult. It's a good, I mean, yeah, of course, it can be think of it, but just imagine, Shruti, like how economical it is. This is the
Yeah,
end
true.
of your DNA, right? So you need to make this analog so that it can go for the competitive inhibition. And making this, it's huge, right? तो
Yeah.
उससे easy क्या होगा? This is like very complicated. ये थोड़ा बड़ा हो जाएगा because you need to target this much area. ऐसा structurally आपको पूरा बनाना पड़ेगा so that you can inhibit the telomerase in the way of competitive inhibition. ये थोड़ा सा complicated है. थोड़ा नहीं, I think it's huge. It's very complicated. What else you can do?
Stop the polymerase activity of telomerase.
Yes,
So that
that
it
is one
doesn't.
thing you can do. So you see, you know that जब हम इसको RNA dependent DNA polymerase बोल रहे हैं, and in the case of the eukaryotes, many proteins, okay, many enzymes, they have got so many subunits. ठीक है? Okay. So when I say it is a RNA dependent DNA polymerase, does it remind you of something else? The activity of another enzyme? I'll repeat, RNA dependent DNA pol है ये, ठीक है? This is also similar to another enzyme. Okay, I'll give you the hint. आ रहा है थोड़ा सा आइडिया? नहीं? Okay, I'll give you another hint. Tanvi?
Reverse transcriptase.
Yes, very good. It is reverse transcriptase. Oh, sorry. Okay. So RNA dependent DNA polymerase is also a reverse transcriptase. So what you can do, you can inhibit the activity of RT. So आप क्या करोगे? Now you can make drugs which is already there in the market against the reverse transcriptase activity of the enzyme, right? So we know that in the case of virus, it is entering into the host cell and finally it needs reverse transcriptase so that it can introduce its or integrate its RNA into the host genome, right? So the same molecule, or not the same molecule, the similar molecule which is targeting the virus can also be used against the telomerase, right? ये एक तरीका हो गया आपका ड्रग बनाने का. Now, what will be the second method or second, mechanism or type you can think of it? This is one. So here you are targeting the enzyme of the enzyme or the enzyme catalytic component of the telomerase. Okay, so this is good. One is done. What about the second mechanism? और सोचो. In the name itself, you can think of it. RNA dependent DNA polymerase. इसी से हमें आइडिया मिल जाएगा. और सोचो. So you have targeted, I'll give you the hint, okay? You have targeted already the protein part of the telomerase. Now what else you can target? Tanvi, questions clear है मेरा?
Yes, ma'am.
Okay, और क्या कर सकते हो? So you know what is telomerase? Rough idea. What else you can target now? नहीं? नहीं आ रहा? No idea?
Target the clamp, sliding clamp.
Telomerase does not have any sliding clamps. Sliding clamp in the case of eukaryotes क्या था? pCNA, which is a different part. Okay, it is not part of any DNA polymerase or telomerase. Telomerase is coming at the end of the replication. So there is no question of having the sliding clamp. Yes. What else? मैं तुम लोगों को ही मजबूर करूंगी आंसर देने के लिए, अदरवाइज यू गाइस वोंट गेट दैट थिंकिंग एबिलिटी. ये श्रुति तुम्हें क्या लगता है? आरएनए डिपेंडेंट डीएनए पॉलीमरेज प्रोटीन हो गया है, आप क्या टारगेट कर सकते हो?
The RNA itself.
Exactly. Yes. So now you are going to target the RNA. So second part होना चाहिए आपका RNA का, ठीक है? Why? Because your telomerase is using the RNA as template. Correct? इसी को यूज करते हुए वो बनाता है आपका parental strand को लंबा कर देता है, so that later the parental strand can act as a template for the daughter strand and then same Okazaki fragment and DNA ligand and वगैरह वगैरह. Right, so I can directly target the RNA. Okay, so kaise? So for this you can synthesize. So you know the sequence of the RNA of the telomerase. Remember what was the sequence? TTA triple G. I told you to remember, right? So you can make a sequence, complementary sequence of this. So what will be the complementary sequence here? A U आ जाएगा, यहाँ C आ जाएगा. Right, so आपको RNA के complementary sequence बनाना है, which you are going to use that as the inhibitor. तो जब telomerase का RNA component complementarily इससे attach हो जाएगा, so it is not available now to act as template for extending the parental strand, right? So this is nothing but also known as your antisense technology. Okay, so antisense oligonucleotides you are going to synthesize. Clear? Clear?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay.
Yes.
Okay, so this is how you need to think. Okay, whenever we are going, जब भी आपका कोई इंटरव्यू आता है, आपसे किसी भी तरह का सवाल पूछ सकते हैं कहीं से भी. So you need to think about it. It's okay, let it be wrong. ठीक है, let it be wrong. It's okay. अभी जैसे for example, she said a sliding clamp about the PC and all. Not the telomerase, but of course, if you can target PCNA, so you can stop the DNA replication. Eventually it is going to inhibit the cell, right? Cell ka proliferation stop ho jayega. So yes, if you want to target the, like the cell should be stopped or prevented, then yes, of course you can target the PCNA. But the problem with PCNA is that it is also involved in other mechanism, normal cell cycle regulation. Correct? So if I target PCNA, it is going to affect the entire normal cell growth also. So along with the cancer, you are also targeting the normal cell. So eventually what will happen? The person is going to die. तो हमें ऐसे सोचना पड़ता है कि कौन से प्रोटीन को हम टारगेट बना सकते हैं, ठीक है? Simple, it should be only affecting the cancer cell and not your normal cells. अभी जैसे for example, we know that आप सबको मालूम है, in the case of cancer, you have seen many cancer patients, those they start losing their hair, right? Hair loss रहता है. Is it only from their head or from the entire body? In the cancer patient, कैसा होता है? So this is happening because of chemotherapy and radiotherapy, right? तो hair loss होता है. So is it only from the head or पूरी बॉडी में से होता है?
I think it's full body, ma'am.
Yes, it is full body, right? Because see, in the case of chemoradiotherapy, it is targeting your stem cells. ठीक है? So उसके वजह से क्या होता है? Of course, it is not directly targeting the stem cells. The main motto is what? To kill the cancer cells. But if you see, the cancer cell has got the ability which is similar to the stem cells, like to proliferate, right? So जब भी कोई stem cell divide होता है, okay, it also divides. एक cell जो होता है again becomes a stem cell and the other one gets differentiated. तो इससे क्या होगा? The population of stem cell is not going to get decreased. It is always maintained. Suppose in your body, let us say the bone marrow, total thousands of stem cells are there. Okay, ये हमेशा पे retain रहेगा. After 30-35 years of age, you will start losing them. But because of the chemo and radiotherapy, the chances of these 1000 stem cells is going to get decreased. और जिसकी वजह से क्या होगा? Hair loss not only from the head, but all over the body, wherever you have got the hair stem cells, they start getting loose. Okay, वो मरने लग जाते हैं. And that's why there will be complete loss of the hair from the entire body. Okay, the basic चीज है यहाँ का. Good. Okay, let's go to the topic now. So what are telomeres? So we know that we are going to just read this thing. So our bodies are composed of more than 1 million cells, and each cells are continuously dying and new cells are continuously being formed. Okay. So inside the nucleus of a cell, our genes are located on the twisted double-stranded molecule of DNA called the chromosomes. Yes, we know that. And all these chromosomes, they have got the telomeres, okay, which protects our genetic data and make it possible to divide and hold. Now, this is also important, right? The advantages of having the telomeres. So already we have discussed. So the entire chromosome has about 150 million base pairs, and each time a cell divides, average person loses around 30 to 200 base pairs from the ends of that cell's telomeres. Okay. Now, this is because enzyme that duplicate the DNA cannot continue the duplication at the end. So already we have discussed about this. Now, cells normally can divide only about 50 to 70 times, which telomere is getting progressively shorter until the cell becomes senescent or die or sustain genetic damage that can cause cancer. Okay. अगर guys आपको जैसे interest है cancer में काम करने के लिए, so you go to a research lab, right? Of course, you are targeting CSR, we will be joining some CSR lab, and जब भी हम किसी lab में जाएंगे, you suppose you're working on cancer or any other mammalian cells, okay, for some other purpose. So always you need to maintain. अभी human cells का क्या रहता है, mammalian cells का क्या रहता है? You know that in 24 hours it is going to divide. Okay, तो आपके पास petri dishes है. Okay, आपने इसमें अपना cells डाला है, and you know that in 24 hours it is going to become double. तो आप स्टार्टिंग में कितना डालोगे? Very little. ठीक है. Now, in the case of mammalian cell, I hope you must have heard about DMEM media. कभी सुना है क्या? या RPMI media, RP. कभी सुना है क्या ये media in animal tissue culture? No. Akshaya, kabhi like you guys have done animal tissue culture?
I have heard of the first one kind of, but I don't remember it exactly.
Okay, so you know that whenever we do animal tissue culture, the plate is having a media of some pink color.
Yes, ma'am.
Yeah, they always have got the pink color ka. In the cell culture, animal tissue culture, there is always this media of pink color only. चाहे आप
Yes,
DMEM लो या RPMI लो, it's actually a indicator because of which it is pink color. So whenever your cells are multiplying, of course they are having those, they are also releasing the growth metabolites. ठीक है, यानी कि उनका waste, the cell का waste, so they are going to throw into the media बाहर. So because of these growth metabolites, the media is going to become acidic in nature, and because of which this pink color will turn into orange, bright orange. So this is an indication that आपका जो tissue जो petri dish है वो full हो गया है. Okay, there is no more media or nutrition remaining now for the cells to divide. अभी आपको क्या करना है? आपको split करना होगा. You have to provide the new media. Okay. So what do you see is that अगर आपने, the main purpose of me explaining this is, when a cell is there, let us say that you had only one, okay, and later after 3-4 days it became orange, like from pink media it became orange. Now once you calculate after 3-4 days, okay, when you're going to calculate using your hemocytometer, etc., whatever method you have, you will see that this cell has gone around 40 to 50 generations only. यानी कि वो 40 to 50 times divide हुआ है, and after that it gets stopped. So one particular primary cell, please remember, huh? In the case of mammalian cells I'm talking about, so primary cell क्या हुआ? What is the primary cell line? Basic. Tanvi, any idea? Primary cell line क्या होता है? नहीं, श्रुति तुम?
Ma'am, yeah, primary cell line is the cell line that is directly taken from the individual and then grown on the media.
क्या क्या? फिर से बोलना.
A cell line that is produced by taking out the cell directly from the individual.
I don't say the cell. We take the tissue directly from the individual and then we homogenize into cells and we make primary cells. Yes. So if you see that any primary cell line which you start after 50-70 times only, it is going to divide. Okay. उसके बाद क्या होगा? The cells are going to die. ठीक है? इसलिए if you work on these primary cell lines, then it becomes difficult. अगर आप किसी cancer cells में काम कर रहे हैं तो there is no problem because they are immortal cell lines. Continuously it can divide, so कोई problem नहीं है. But if suppose your research work is on the primary cell line, then every time you need to take the tissue, every time you have to start the experiment from the very beginning. So that's why it is what to say, difficult line of research. Like if suppose you lose the cell or because of some drug you added, the cell is dead, then again you have to start from the tissue. So that's why it becomes little difficult. Okay. Yeah. So yes. Now we know that why the cell is dying, because the telomeres are getting short and later they die because of the apoptosis. Okay. Now in human blood cells, the length of telomeres ranges from 8,000 base pair at birth to 3,000 as people age. Telomeres do not shorten with age in tissues such as heart muscles, in which cells do not continuously divide. So here again, so we know that the heart cells are not dividing, and that's why their telomeres are not getting shortened. Okay, these are the few things which you need to remember, because based on these sentence only the questions are being framed. Okay.
Ma'am? Even in brain the cells don't divide.
Yes. After certain time, yes, after certain age, the neurons stop dividing. So there also, yes. Now the thing is like, अभी आपसे अगर ये अभी आप पूछ सकते हो कि अगर हार्ट सेल्स डिवाइड नहीं हो रहे, न्यूरोनल सेल्स डिवाइड नहीं हो रहे हैं, राइट? सो कैसे इट इज मेंटेन, लाइक हाउ? Are you getting my point? अभी जब वो डिवाइड ही नहीं हो रहे हैं तो just imagine how much work is there, workload is there on these existing heart muscle cell and the neuronal cells, right? इसलिए you have to have a healthy life after 30-35, because you're not going to have the division of these cells. Okay, अभी कुछ damage होगा, so what will happen? If any accident is happening or you're getting a heart attack, so what will happen? Now there will be the loss of these existing cells, and hence the lifespan shortens then. ठीक है? तो जब तक आप छोटे हो, you are like in the teenage and all, तो आप कितने बार भी गिरो, कितने बार भी हड्डियां तोड़ो, everything is fine, you will be healed properly. But after 30-35, you have to take care of your health, and that's why people say ना कि आप हमेशा exercise करते रहोगे, so it is good, because these exercise things are releasing the good antioxidants which will keep your neuronal cells, heart cells in healthy environment. Okay, now we are just getting moving to the different topic itself. Okay. So this is just the cartoon representation of the same. So you know that this is your first young cell. Okay, so as it keep on dividing, okay, this is not getting replaced. Okay, so what will happen? You can see that now the telomeres are not there. So you will see that in the case of the old age people, the telomeres are almost not present and those cells are dying eventually. Okay. Now look at the graph. This graph is important. Okay. So this is the number of cell division versus number of nucleotides. So here on the y-axis, you have got the nucleotides, and on here x-axis, you have got the number of cell divisions. Okay. So Akshay, what do you understand from this graph? You can see that as the cell division is going, you can see the graph is like this. Okay. So what is the conclusion over here? Let us say that this is 1, 2, 3, 4, 5, and 10, 5 and 6, and each cell division is happening after 10 years, so like something like that. So ये 60 years का दिया हुआ है. Just imagine like that. Yes. So what can you conclude?
Number of that the telomeres are getting reduced and reduced, so the number of nucleotides is also gradually decreasing. That's why we age.
Yes. So that's why we see that there is a falling slope over here like this. ठीक है, नीचे की तरफ गिर रहा है. So with the increase or with more number of cell divisions, the number of nucleotides in the telomeres is keep on reducing. Okay, good. I mean, it looks very simple now, right? Graph is looking very simple, but the question आता है थोड़ा सा confusion आ जाता है. Anyways, so how your telomere structure looks like? So they are composed of the repeated sequences. Okay, and they have got the telomeric interacting proteins. Now, this telomeric interacting protein is also a very important concept, and they are known as shelterin. Have you guys studied about shelterin in your college, BSc, MSc? Yes.
Yes, ma'am.
So
Yes, ma'am.
okay. So what is the main function of shelterin? There are two main functions. What are those? Yes, Shruti?
And they actually cap the telomeres so that the cell doesn't repair the telomeres like it repairs everything, all the other single stranded DNA. And it also
because the telomere is looking like a double stranded break. So it is going to cap. Otherwise, it will undergo the repairing mechanism. तो क्या होगा repair होगा तो इसको ऐसे जोड़ देंगे, which is not correct, right? Okay. What is the next step? Next application.
It also, I think it surrounds the cap because it makes this D-loop structure. So, and then it caps the rest. I'm not very sure about it. It was in first year.
Okay, so what you said is this is correct, that it is capping the telomeres. Okay, so this is happening. Okay, anyways, it is there in the upcoming slides. It's okay, we'll discuss here itself. See, as we know that the telomere DNA is thought to adopt a T-loop structure. Now, this T-loop structure is why it is coming is because of these sheltering. जो भी हम देखो पढ़ेंगे, the structure of the telomere, you will see that it has got T and D loop, the displacement loop is there. Now, why it is there? Because of the sheltering. Now, sheltering का first काम क्या है? To cap the telomeres, because I know it is going to it is helping in the prevention from the DNA repair enzymes, okay, because it will look like a double stranded break. So it is capping the telomeres. Second is, in the case of the stem cells, okay, embryonic or the hematopoietic stem cells, it is recruiting the telomerase. ये इसका second function है. First क्या है? In the somatic cells, it is going to cap the ends of the telomeres. Okay. Second function क्या है? To recruit the telomerase. अंतर समझ में आ रहा है? Shelterin is present in both the somatic as well as the stem cells. Stem cells may, it is also helping in the recruitment of the telomerase, and in the case of the somatic cells where the telomerase is absent, it is helping in capping the telomers. इतना clear है?
Yes.
Okay. Okay, so this is the structure. You can see that we have got the overhang over here, right? So what kind of overhang is this is called? If you see the structure, that's PTA, G, PTA, triple G is there. So what kind of overhang this is? 5' overhang or 3' overhang? यस तन्वी क्या लगता है? अरे तो क्या अगर गलत हुआ तो मैं मारने थोड़ा ना आऊंगी. It is there in the front. I just want see. I'm asking these questions because it tells me how much basic understanding is there. Like आपको कितना समझ में आ रहा है? I have not asked a hard question. Okay, Akshaya, what do you think? What kind of overhang it is? You have got 5 dash over here and 3 dash over here. Overhang मतलब क्या होता है? अगर यहाँ तक होता तो this is kind of blunt end जो हमने मोल बायो में about the restriction enzymes में पढ़ा होगा, right? If I have got something like this, sorry, कुछ ऐसा दिखता है तो we always say that it is sticky ends. Okay. Now what kind of overhang it is? यहाँ पे आपको क्लियर दिखाई दे रहा है, यहाँ पे 5 डैश है, यहाँ पे 3 डैश है, तो ये किस तरह का ओवरहैंग है? ओवरहैंग मतलब एक्स्ट्रा लटका हुआ, राइट? So this is what kind of overhang?
3 dash.
Yes, it is 3 dash. Simple. Okay, I was just checking the confidence about like how much you have. सबको लगा था 3 dash overhang है? Everyone thought that this is 3 dash overhang.
Yes, ma'am.
तो बोलना है ना, be confident, okay? My motto is, along with the theory, you should be confident for the interview. Frankly, guys, anyone can clear CSAI. सिर्फ इंटरव्यू क्लियर करना बहुत इंपॉर्टेंट है आजकल, बिकॉज एवरीवन नोज द कंपटीशन इज हाई, इसलिए इंटरव्यू क्लियर करना बहुत इंपॉर्टेंट हो गया है. एंड मेरे टाइम पे तो सिर्फ 1 राउंड ऑफ इंटरव्यू था, नाउ इट्स लाइक 3 राउंड्स ऑफ इंटरव्यू एवरीवेयर. Okay, no problem. Just be confident. Now, that's the structure of the telomerase. Can you see? Are you able to locate the RNA over there? Can you see along with the protein alpha beta, you can see that there is a RNA structure also. Can you see this purple color wala? Okay. So we know that, okay, does anything counteract the telomere shortening? Yes, it is telomerase. So it is going to add the pieces to the ends of the telomere. So in the young cells, telomerase are there. So they help in, keeping the division good as it is. Now, they grow shorter as the cell is going to age. Now, telomerase is turned off in the somatic cells, but remains active in the sperms and the eggs, which is passed from one generation to the next, and also in the case of your stem cells. Okay. Now, telomerase activity is lower absent in the normal cells and all, etc. So this will not be wrong. Telomerase. Now it is important. This point is important. That it is composed of catalytic subunits HTERT and HTR, where H stands for human. Okay, H stands for human. Now let's see the structure. Okay, this is the structure. This is nothing, guys. You can just read also later. So we know that it's a ribonucleoprotein. So why it is ribonucleoprotein? Because it has got the RNA as well as the protein. It has got the 3' overhang. Okay. Now, telomeres protect the end of the chromosome from the DNA damage, etc., etc. पहला study हमने Drosophila melanogaster में किया. Now, this is the sequence in the case of Trypanosoma brucei. So there is another slide which I had kept for you, where you can see the sequences of the Trypanosoma में कैसा दिया है और दूसरे brucei में कैसा दिया है, and humans में कैसा दिया है. I can show you now. हाँ, yeah. Sorry, Trypanosoma brucei एक ही है. So here you can see the sequence. Okay, आपका tetrahymena thermophile में सबसे पहले हुआ था. You can see the sequence similarity. TT double G TTA triple G. Okay, तो आप देख सकते कितना similarity है. Here also you can see triple C AAU triple C. This is again, अगर आप यहाँ से देखो थोड़ा बहुत तो similar लगता है, right? This is like TTA triple G only. अगर हम देखें तो, है ना? But this difference क्या है? This is 5' 3', but ये आपका 3' 5' है, and again this is 5' 3' है. थोड़ा सा अंतर पता चला? Even though the sequence is same, the direction is different. Okay, Now the difference in the direction is making telomerase also different in all the species. So from this, you should understand that the telomerase is species specific. Okay, ये point clear हो गया? Now why I am saying this, giving more stress to this point is because the question was there, whether the telomerase is species specific or not. Okay. अब इससे वो चीज प्रूव हो जाती है. Is this much clear to you guys?
Yes, sir.
Okay. So let's.
Ma'am?
Yes. Yes. Someone was asking question.
Yeah. But then there are large groups of organisms that have the same sequence and therefore probably the same telomerase, right? Because all mammals have exactly the same one. So
Mammals
is it?
are species specific, I said. So human species is going to have same kind of telomerase.
Yeah, but then dolphins and humans also have the same telomerase as well.
No, the difference will be there. Dolphins are coming under the category of mammal. No, not mammals. I forgot. Dolphins comes under mammals only, because they also lay.
Yeah, they do.
Okay, so no, I mean, if you ask me like this is also confusing for me, but no, I guess dolphin is not coming under the human species, but right?
Yeah, but they have the same telomere. The sequence is the same though.
You are telling me or asking me?
I was just trying to say that a large groups of animals generally have the same telomerase structures. So is it species specific or like, because a large group of organism has the same telomere? I was not sure about the species part because large groups have the same
when you
telomere.
say large group of organism means okay, so see what I studied and what I understood is that when I say species specific, okay, humans comes under one species, let it be from any continent, okay. Then aquatic animals, they have got the different, but I don't think they will come under the species. Okay, we all are mammals, but we are not coming under the same category, right?
Okay.
है ना? वहां पे मेरा ये है, like the point is that when I say species specific, the difference will be there in the east, the difference will be there in the like that I'm saying the species specific. Maybe we can.
Yeah, because I had read that the telomerase sequence is conserved in large groups of species.
No, when you say telomerase sequence is conserved, so those domains are conserved. जैसे कि जो आपका reverse transcriptase का जो activity का जो domain है, that is conserved. Like that you can say. See, that's why there is one software, maybe I have mentioned, I don't know about the PyMol. It's a free software actually. Okay, so it's a free software. आप उसको download करके उसमें जो भी आपके रहेंगे, like for example, the proteins which are present. Okay, so those you can see the structures over there. Okay, guys, am I audible to you?